comes from a background in philosophy and cognitive science. He is interested in the relationship between mind and gray matter. Specifically, his work aims to characterize how the capacity for cognition and consciousness arises from the complex interactions between brain systems viewed as a dynamic network. To this end, he combines tools from information theory, network science, and whole brain computational modeling to study brain structure and function throughout a variety of states of consciousness, such as anesthesia and disorders of consciousness, but also sleep and psychedelics. Ultimately, Andrea aims to develop a landscape of altered states of consciousness and a unified theoretical understanding of how consciousness is lost and how we can promote its recovery in patients. And so with that, Andrea, please take over the screen share. Thank you.
Can you hear me now? All right, fantastic. So you should be able to see my presentation now, hopefully. And it should be in presenter mode now. Is that all correct?
Yep.
Fantastic. All right. So thank you so much for having me. Yes, so I'm Andrea Luppi from the University of Cambridge, and I'll be talking to you about the dynamics of brain integration and segregation under LSD. And I work in the cognition and consciousness imaging group at Cambridge. And our interest is in, as the name suggests, understanding consciousness, human consciousness. And to some people, this might seem almost a paradoxical endeavor, because as William James famously said, consciousness is like the flights and perchings of a bird. So how can we possibly understand that? And yet, neuroscience has been making some great strides. And we now have computational and mathematical models of how cognition and the brain might work at a very high level. And in particular, this is the Reboost model by Carrot, Harris, and Friston. And I know there has been a prior presentation on this topic, but I'm just going to introduce the part of this model that is at the core of this particular work. And the Ribose model says that under normal conditions, your perception, your experience is effectively the interpretation that your brain makes of the sensory inputs that it receives from the environment. And the reason this is important is because it means that your prior expectations are shaping your experience. So for instance, if you have spent all of your life looking at people's faces, like most of us have, then you might find that these things that have actually no face in them do look nevertheless like faces. And there comes the second part of the Reboost model, which is that Carrot, Harris, and Friston believe that under the effects of psychedelics, one of the main ways in which cognition becomes different is that it becomes less constrained, because psychedelics weaken these top-down effects, or as they say, they generate relaxed beliefs under psychedelics. And the point where we come in is that these are predictions that we can actually test empirically using neuroimaging data. And the approach that we take in particular is through the field of network neuroscience. And here it is displayed, which is to say, we use functional MRI and other neuroimaging tools to obtain data from the brain. And then we turn brains into networks because we have a very highly developed science of networks. This is called graph theory. And we can use it to obtain insights about those networks and then turn them back into insights about the brain. And we're all familiar with networks because this is what we actually use to navigate in the world. Our world is full of networks all around us, including even social networks. But I will focus here on transportation networks as a particularly useful example for the purposes of this presentation. And in particular, if you think of a transportation network, you can distinguish two separate aspects, which is to say you have the structure, which is the roads that connect different cities, but you also have the function, what is actually happening on those roads, which in the case of a transportation network would be the traffic. Now, this turns out to be an actually quite useful metaphor for how the brain itself works, because you can think of the brain as being divided into many different regions corresponding to the cities in your network. And these regions are connected by physical connections that play the role of roads. And on those roads happens brain activity. And you can ask, how does brain activity work over time? How do different regions talk to each other? And through neuroimaging, you can actually obtain representations of this, as you can see here. And these are called matrices, and you can think of them as maps of brain interactions, function and structure, respectively. And as you can see here perhaps more clearly, there is a high degree of similarity between structure and function, structure being here on the left, function being on the right. But this similarity is not perfect, nevertheless, which is to say the roads influence what traffic exists, but they do not fully determine it. And one question that you can ask at this point, thinking of brains, is how do brains connections, the roads between brain regions, how do they get there anyway in the first place? And to a first approximation, I would say that you can have a two-part answer, which is to say, in part, they are coming from evolution. So here on the left, you can see the connectivity, the roads between brain regions of a macaque at the top and of a human at the bottom. And you can see that although there are clear differences, there are also very clear similarities, like the checkerboard structure of them, which is to say, the way that the brain regions are connected is partly determined by evolution. And on the other hand, it is also partly determined by your own personal history of learning. Because as Donald Hebb famously said, neurons that fire together wire together, which is a memorable way of saying that whenever two neurons talk a lot with each other, the result is that the brain will create a highway between them to facilitate this communication. And eventually this will be reflected in the presence of connections between different brain regions, for instance. So your individual connectivity is shaped by these two separate aspects, evolution of the species, but also your own personal history and learning. So brain structure is very much plastic over long timescales. And at this point, you might be wondering, why does this all matter? I thought this was a presentation about psychedelics. And it is. Because the reason I've been talking about brain structure and function is because you can think of brain structure as representing the brain's prior expectation about which regions should be talking to each other functionally, which is to say, the presence of a road should predict whether those two, the corresponding regions that are connected by that road, are talking to each other a lot or only a little. In other words, structure represents a prior expectation on function, and that means that this is something we can look at with neuroimaging to test the predictions of the Ribose model of psychedelics. So here come the psychedelics again. Because now we have a testable hypothesis. Do psychedelics weaken the prior constraints that we see in the brain in terms of the effect of brain structure on constraining and informing brain function? However, before we can actually test that, there is one more aspect that we need to take into account. So let's think again to traffic network. Traffic is not always the same over time. And in particular, as this graph here shows, during weekdays, when people need to congregate in the same place to get work done, you see a lot of traffic. On the other hand, during weekends, people are able to stay in their own homes and act independently on their own business. And as a result, there will be much less traffic, which means in turn that even though the roads may be the same, their relationship with the traffic that is taking place on the roads is going to be different over time. So unlike structure, function is very much time dependent. And it turns out that this is not just about the network of traffic happening over roads, but it is also true of the interactions of brain regions taking place over the brain connectivity, which is to say, you can find over the course of even just a few minutes, you can find moments in time when the brain is in a more integrated state, where brain regions are talking a lot to each other. And you can find also moments in time when the brain is in a more segregated state, a bit like the weekend in traffic, which is to say the brain regions tend to only talk to their own neighbors and not so much at a global level. And of course, we can image both of those during the psychedelic experience. And we can ask, specifically in this case, in the context of LSD, how does LSD change the way that the brain communicates during these integrated and segregated states? So here comes the first part of the results of our study, which is to say, our first result was that during the segregated state specifically, we find that the brain is exhibiting a more diverse pattern of functional connections. That is to say, the connections between different regions of the brain are more complex and less predictable. And this is interesting because it goes in exactly the direction predicted by the entropic brain hypothesis, which is a precursor to the Ribose model. And it predicates that under the effects of psychedelics, such as LSD, you should see higher entropy, higher diversity in brain function. And this is exactly what we find, but specifically to this segregated state of brain function. Now, an even more direct test of the hypothesis that we had set up to investigate is that we can look at the similarity between brain structure and brain function. Because I've been telling you earlier that these two maps, these two matrices, look like each other, but we can actually measure how similar they are. And what we find is that, again, only during the segregated state, LSD reduces the similarity of brain structure to brain function, which is to say, exactly as we had expected, under the effects of LSD, this structural constraint, structural prior expectation on brain function becomes weakened. And finally, the third result that I want to talk to you about is again specific to the segregated state. And this result is about this particular brain region here that I am highlighting in a circle. And it is a brain region in the front of the brain that becomes disconnected from a number of other regions in the brain. As you can see, there are very many changes in connectivity during the segregated state that are induced by LSD. Some regions become more connected, some regions become less connected. But the reason I am specifically focusing on this particular region in the front of the brain is because this region has been implicated with the so-called reality monitoring process, which helps us to understand whether what we are experiencing and what we are remembering is real or is instead just imagined, and it has an internal or an external source. And I think this is very relevant to CJ's own presentation from earlier. And this region has also been shown to have an abnormal pattern of connectivity in patients suffering from schizophrenia, especially those who suffer from hallucinations as a result of this condition. So we can think of this particular region as helping the brain in some sense to determine whether our expectations are real or not, which is another way to say that it is exerting a top-down control over the experiences that we have based on our expectations. And this is just to show that that region is in fact the region as the paper I was mentioning is referring to. And there is quite striking overlap, if I can say so myself. And so to summarize these results, we obtain three separate aspects that are all going in the same direction, which is to say, we find that there is less relevance of the structural priors on brain function under the effects of LSD, which is what the Ribose model would predict. It would predict less effect on prior expectations on brain function. At the same time, we also find that there is a more diverse pattern of connections, less predictable, under the effects of LSD. And finally, we find that under the effects of LSD, the brain is less able to determine whether its experiences are real or not, which is to say, the key region implicated in reality monitoring becomes less connected with the rest of the brain. And the important thing is that all of these effects are found at the same points in time because they are specific to the segregated state, which is telling us that it is important to consider these brain dynamics in the process of understanding the effects of LSD on the brain and presumably also the mind. And this is especially relevant because many of these effects are the opposite of what we see in other states of altered consciousness, namely anesthesia, when instead consciousness is entirely lost. And at this point, it is important to emphasize, though, that the Reboost model is much richer than what I have just been described. As I said, there has been an entire presentation dedicated just to this. And in particular, although here we focused on testing the prediction of weakened priors using neuroimaging, the same prediction can also be tested through a different avenue in terms of a behavioral weakening of the priors. And this is some of the work that John Kenyon and others have been conducting, and I would very much encourage you to check out this work. And on the other hand, the Reboost model also makes predictions about a so-called flattening of the energy landscape. And for reasons of time, I am not going to describe in detail what this means, but suffice it to say that this is related to the question of this weakening of the top-down priors. And this can also be tested empirically through neuroimaging methods. And this is precisely the work that Parker Singleton, who I think may be attending this very call, is conducting to try and test this additional hypothesis once again empirically using neuroimaging data. So I would recommend checking out these great guys' work. And finally, this is just as like with all of my references, which I believe will be made available to everybody, so you can check all of these papers that I've been mentioning in a lot more detail than I could be mentioning here. And this is just to say that the paper that all of these results are based on is itself available open access in Neuroimage, so you can check it out. And finally, I really want to take the time to thank a lot of the people who have helped in conduct all of this work, but have also helped more broadly with my PhD and my research so far. And first among them are, of course, my supervisor, Dr. Stamatakis, and my advisor, Professor David Menon from the Cognition and Consciousness Imaging Group in Cambridge. And you can see here the other members of the group, but there are also additional collaborators in the University of Cambridge and even more collaborators beyond Cambridge itself in the rest of the world. And first and foremost among them for the purposes of this presentation are, of course, Dr. Robin Carter-Harris and Dr. Leo Roseman, who actually collected the LSD data that I've been talking to you about, which was a huge effort. And they were generous and kind enough to share those data with us so that we could together explore these results and eventually come up with this paper. So huge thanks to everybody who has been contributing to this, and including my funders, Gates Cambridge. And of course, thank you for your attention. And I'll be happy to take any questions that you may have.
Great work. Thank you so much, Andrea. That was a fantastic presentation. You took some really complex topics and distilled them with some awesome analogies. That was fantastic. So again, if anyone has questions, please ask them in the chat. If we don't have any right now, oh, we've got the first one here. Okay, this one's from Carly, and they are asking, did you say the task for the participants recorded? Is there a behavioral distinction, or is the task for segregated versus integrated states in the study introspection / mind wandering / sitting versus external engagement, navigation, visual exploration? I don't know if you got
that. So yeah, so I did not actually mention that, but I should have, which is to say that these are resting state data. So the participants were not doing any task in particular. They were just lying in the scanner, and they were asked to not think about anything in particular. And we were recording their brain activity as they were lying there in the scanner. And so there was no specific task. What we do know, however, is that the integrated and segregated states have different relationships to different kinds of tasks, because this was studied in other work, including the Shine paper that is among my references. So we know that the integrated state is more related to higher cognitive function, like working memory, whereas the segregated state is more related to relatively low-level sensory motor tasks. But it is important to emphasize that during our recordings, the participants were just in the resting state without doing any specific tasks.
Perfect. We got another question here. And the question is, were the brain regions most affected by LSD the richest in the 5-HT2A receptor expression?
Right. So we do know that LSD influences 5-HT2A receptors in a rather specific manner. In this particular case, we did not explore the relationship with 5-HT2A expression in this study, although for that there are some great papers using the same data and using, for instance, 5-HT2A receptor information obtained from PET mapping to identify the role of this receptor in generating the kind of dynamics that you would observe under LSD. In our specific task, in our specific study, what we did show, although I didn't present it here, is that there is a loss of anticorrelations in the brain, and we know that typically those involve the default mode and frontoparietal networks, and we know that those also tend to be rich in 5-HT2A. So I'm confident to say that there is an indirect effect of 5-HT2A specifically, because we know that that has been already investigated in our data, but we did not specifically investigate this in our study.
Okay, great. Another question here. Do changes in functional connectivity lead to changes in structural connectivity over time in terms of neuroplasticity due to LSD consumption?
So for accepting the part of your question that is about due to LSD, we do know that over long time scales, the brain function is itself going to shape brain structure because of plasticity, which is to say, we know that, for instance, if you learn to juggle, I think this was a famous study, people learn to juggle, and as a result, they found that their brains were changing and they were observing changes in structural connectivity. This being said, at the moment, I am not aware of any evidence indicating that LSD would change brain structural connectivity. So we do not have, as far as I know, evidence about that.
Okay, thank you. Another question coming in from Carmen. They're asking, will you publish another paper using the same LSD data in the future? And what would you explore if you were to publish?
So the answer to that is, in part, we have already a preprint about these data, which is looking at the relationship between different psychedelics, so LSD, but also sub-anesthetic doses of ketamine, so at dissociative levels, comparing them using the same analytic framework to relate them to anesthesia and also disorders of consciousness. So we're trying to develop a landscape, if you will, of these various states of altered consciousness. And we are finding some interesting similarities between anesthesia and disorders of consciousness on one hand, and ketamine and LSD on the other hand. And those tend to be mirror reverse images of one another. That paper is currently available as a preprint on bioRxiv, and I'll be happy to then send the organizers a link for that.
Perfect, and yeah, we'd be happy to share that too. Another question here coming from Keith. What does it mean to have a negative participation coefficient?
So the participation coefficient is reflecting the extent to which a brain region or any node in a network is interacting with different modules of the network. So if you take a network and you can divide it into modules, which are large-scale communities, you can think of them a little bit like cliques in a high school circle of friends. And in that case, the participation coefficient is telling you, is this region or this node in the network interacting with many members of other modules, or is it instead interacting only with members of its own modules? And the more members of different modules it interacts with, the higher its participation coefficient will be.
Perfect, thanks. There are no questions right now in the chat, but I do have a question for you, Andrea. I've heard a lot, and when you're talking about brain models relating to psychedelics, it's always the Reboost model that's brought up. Are there currently any other models in the literature that challenge or are significantly different from the Reboost model currently?
So that is an interesting question, in the sense that often the word model is underspecified, which is to say that, yes, there are many different models, but of consciousness in general, and some of them also specific to psychedelics. But they tend to be often specified at different levels of understanding. So for instance, some models might focus more on the effects of specific regions or on the role of specific receptors, for instance. Whereas at least the aspect of the Reboost model that I've been talking about here is mostly focused on a relatively high level understanding of brain function, which is derived mostly from the combination of, for instance, free energy framework and then at the same time, Kara Harris's entropic brain hypothesis. And each of them is specified at multiple levels, which is to say that, for instance, Kara Harris's model talks specifically about the DMN in quite a lot of detail. But the question of how to bring them all together, and in particular, whether they are actually conflicting or not, that is difficult to answer in a definite way, precisely because often they're talking about slightly different aspects of what it is, what effectively slightly different things that they're trying to explain, and as a result, slightly different entities that they can take as doing this explaining. And I should add, this is by no means specific to psychedelics. This is very common in theories of consciousness more broadly as well, which is where most of my familiarity is with. So many different theories of consciousness exist, and often they talk about slightly different things when they say consciousness. And some of them more focused on neuroanatomy, some of them more focused on things like information theory, for instance.
Perfect, thank you. I don't think we have any other questions as of now. So what I'm going to do is say, first of all, thank you, Andrea, for your amazing presentation and sticking around for the Q&A. And I'm just going to pass things off to Leo to finish things off.
Whoops. Okay, thank you, Aiden, and all of our speakers, Andrea and CJ, for giving us this wonderful presentation today. Just for some housekeeping, our next our next meeting will be May 20th, and it will be at the same time as today, because Nadia will also be coming from Europe, not sure which country, and so we'll be starting a little bit earlier as well. And of course, we have our own Michael Hachen coming to discuss the antidepressant effects after discontinuation of MDMA-assisted therapy as well. So thank you guys all for coming to our meeting today, and we hope to see you guys next month. Take care. Bye-bye.